think that we can start. So, good afternoon to everyone. It is my absolute pleasure to introduce today our speaker, Professor Rahm Gummuluru, whose work stands at the forefront of our understanding of virus-host interactions and innate immunity and therapeutic innovation. I first met Ram many years ago in Costa Rica Retrovirus meeting, and I'm very happy that you are here at the Institut Pasteur today. So Professor Rahm Gummuluru began his academic journey with a Bachelor of Science degree from the University of Saskatchewan in Canada, followed by a PhD from the University of Rochester in Rochester, New York State. Next, Ram was postdoctoral fellowship at the Fred Hutchinson Cancer Research Center. Currently, Ram is professor in the Department of Virology, Immunology, and Microbiology at Boston University School of Medicine, investigator in the National Emerging Infectious Disease Laboratory, and co-director of the developmental core for the Lifespan Boston University BNC Center for AIDS Research. He leads a dynamic and multidisciplinary research laboratory. His team focuses on macrophage pathogen interactions, an area that intersects cell biology, biochemistry, nanotechnology, induced pluripotent stem cells, and animal models. His work has significantly advanced our understanding of HIV-induced immunopathology, particularly mechanisms of viral entry and innate immune signaling in macrophages and dendritic cells. Over the years, his research has broadened to include enveloped viruses beyond HIV, such as paramyxoviruses, filoviruses, and coronaviruses, deepening our insight into innate immune sensing in myeloid cells. Notably, Gummuluru's group is also pioneering the use of nanotherapeutics to target virus reservoirs in tissues, offering promising avenues for tackling persistent HIV infection in CR-suppressed individuals. So, Professor Rahm Gummuluru, please join me, and the floor is yours.
All right. You can hear me, right? Okay, fantastic. Thank you, Francesca. Thank you for the kind words. Thank you for the invite. I was delighted to come to Pasteur because I wanted to come to Paris. I was in Erlangen, Germany for a visit, and I asked Francesca, hey, can I come down here, please? And she said, yes. I was like, okay, thank you. So I'm here. And so I'm going to spend about the next 45 to 50 minutes trying to tell you about two stories, one published, one going through the rounds of reviews. So happy to take some feedback on both of them as we go forward with it. All right, so before I go forward, I think Francesca briefly gave you an overview, but what my lab does is we're interested in questions about pathogen interactions with macrophages, and we are in many ways agnostic to the pathogen. We're interested in HIV, we're interested in Ebola virus infection and innate immunity to Ebola infection, SARS-CoV-2, as was true for the whole world about a few years ago, and we continued in that vein as well. More recent work is now focused on malaria as well, and we're specifically interested in question of heterologous immunity in endemic malaria infections, especially in sub-Saharan Africa. But the talk today is going to be focused on innate immune sensing and the consequences of persistent innate immune activation in people with HIV. And the reason this is of interest to us, and it remains a major issue, is even though we have excellent pharmacological options, and ART has dramatically reduced overall mortality rates in people with HIV, there still remains a clinical burden, and that most of these ART-treated individuals still progress to end-stage disease from non-infectious causes, right? So we call them HIV-associated non-AIDS complications. So these are, in many ways, especially evident in aged people with HIV. So you see complications due to cardiovascular dysfunctions, neurocognitive dysfunctions, diabetes, cancers. These are all accelerated in these individuals, and there has been an underlying theme that defines all of these complications, and that's been immune activation. And these are numerous studies that have pointed this correlation. I'm just highlighting one of them here. So all these studies seem to point out that chronic immune activation and inflammatory markers are associated with greater risk of non-AIDS morbidities. So what is the driver of this? And this is known for a while. Chronic inflammation is the driver. But what induces that inflammation, that persistent inflammation, is still a matter of debate. So this has long been even observed in the gold standard animal model, the SIV-MAC infection of rhesus macaques, right? So where there is sustained upregulation of type 1 interferon responses and expression of ISGs. And this was highlighted from a study, and there are other studies. I just chose to highlight this study from Guido Silvestri and Steve Bossinger, where they compared the infection course in natural hosts versus a pathogenic infection. And while they're very similar in both cases, while replication set point is achieved when viremia and then viremia persists, what is differentiating between the two is the persistent ISG expression that persists for the duration of the pathogenic infection, right? So this chronic inflammation that is not observed in the natural host. So the resolution of inflammation is key. So our questions in HIV come from this argument that there are these persistently infected sources, and we have focused on macrophages as the key pro-inflammatory mediators in this. So macrophages make up part of the reservoir. They clearly are not the chief players. The chief players are CD4 positive T cells. But about approximately 4% of all infected cell populations do contribute to this reservoir in vivo. And they're persistently infected in various tissue sites, brain, lung, secondary lymphoid tissues. One of the ones that we also work with in the lab is microglia, and that have been implicated in CNS impairments. Okay. And what is also well appreciated is that the monocytes from people with HIV display a sustained pro-inflammatory phenotype. So our question that I'm going to ask today is that whether there's persistent expression and sensing of these pathogen-associated molecular patterns in macrophages drives that inflammatory phenotype. And one of the questions that remains and is still a question that we are actively interested in is whether this also contributes to in the activation in the context of a latent reservoir that remains transcriptionally active, especially even defective proviruses, which constitute over 90% of the reservoir in ART-suppressed individuals. I'm not going to answer that question to you today, but this is an area of active research in my laboratory and my collaborators, Andy Henderson at Boston Medical Center, where we're interested in questions of how defective proviruses also promote persistent inflammation. Okay, so the work itself got started by this experiment approximately 7 years ago now, 8 years ago. And this was a finding that we were consistently observing at that time. So these are peripheral blood monocyte-derived macrophages, which were infected with a replication-competent HIV. In this case, we used an YU2 isolate. And we compared the intracellular P24 GAG as a marker for viral replication in these cultures. And when we compared the two cell types, the uninfected versus infected, we reproducibly saw upregulation of a protein called CD169 on the cell surface of both infected cells and uninfected cells. And these were the days we were interested in cell-to-cell transmission, and CD169 also is called Siglec-1, which is a sialic acid binding protein that is crucial for cell-to-cell transmission of HIV. But what was surprising to us was this upregulation on the surface of both infected and uninfected cells upon HIV infection establishment. So why was this interesting? It's a type 1 and type 3 interferon inducible protein, right? So this is an expression which is normally not constitutively expressed at high levels, but if you were to expose these cells to interferons, you will get high expression. Turns out it's a very nice ISG to use and very useful in the laboratory. Its expression can be upregulated in vanishingly small amounts of interferons, type 1 interferon. So this is like 1 unit of type 1 interferon where we already see about 5-fold induction of this expression. So nice marker to track activation status of myeloid cells because it's only expressed on macrophages, microglia, only myeloid cells in vivo. So we did that, and we also looked at other ISGs. In this case, we looked by RT-PCR for type interferon beta message in these macrophages. And we found another interesting phenotype, which was that this induction was not observed at early times post-infection. This is day 1, day 2 post-infection. This was induced pretty late in the virus life cycle, at day 3 post-infection, which was suppressed if we had an RT inhibitor in the system. Okay, so this was a late step in the virus life cycle that was triggering these responses in the infected cultures. This is not only true for type 1 interferon beta message, it's true for other ISGs that you want to look at. We looked at IP10, ISG 15, 54, 56, Viperin, MX1, and the pattern remains the same. You see late induction of these messages, some much higher than the others. IP10 is an excellent marker for this. And in all cases, an RT inhibitor would block inductions of these ISGs in these cells. So a replication established, productive infection establishment was required for this phenotype. So this is not a new story, at least in HIV and macrophages and HIV innate immune activation. There have been lots of investigators, some of them are in this room, who have been interested in this question of what drives innate immune activation in macrophages. And there have been lots of different steps which have been postulated to promote this activation step. There is data that's published that argues for fusion pores, capsid encoding, and TRIM5 alpha as a pattern recognition receptor that recognizes an intact capsid, reverse transcription intermediates, which are somehow exposed in the context of maybe suboptimal infection conditions or deficiencies in capsid encoding, which then triggers cGAS sting, nuclear import itself, which MX2, which blocks it, double-stranded DNA breaks or DNA damage responses, which have also been argued for inducing innate immune activation. What I'm going to show you today are two steps, which are relying on the late viral gene expression, as well as viral assembly, which we think are contributing to this event that I'm going to describe to you today. And the reason we are confident about this is that if we take macrophages and we do the same experiment that I just described to you, but add inhibitors that block virus entry, viral reverse transcription, viral integration, viral transcription, or protease activity, we find the phenotype that I just described to you, that any of the early steps which are blocked all the way to viral gene expression block CD169's upregulation on the cell surface. Once infection has been established, once there is gene expression or protein expression, you see upregulation of CD169, okay? So this was our way of saying that we're confident that this was a late event in the virus life cycle. And this is not only true for CD169, we could also see this for IP10 and other pro-inflammatory markers, same phenotype. This is work that was initiated by a postdoc in the lab at that point, Hisashi Akiyama, who's now independent and has his own laboratory interested in other questions about RNA sensing. So when Hisashi set this up, he also tested this in another cell type that we are very interested in. These are iPSC-derived microglia. These are microglia that are derived from pluripotent stem cells, so we can model tissue-resident macrophages. And again, the same phenotype holds for these cells as well. Replication establishment or prevention by RT or integrase inhibitor or a gene expression inhibitor blocks induction of IP10 or MCP1. So diverse cell types. This is also true for dendritic cells. This is also true for CD4 positive T cells, which is not our data, but data from Jeremy Lubin's lab. So this seems to be relevant in all virus permissive types in vivo. So we immediately focused our attention on the viral RNA or viral protein. That was the driver of this response. I won't show you that data, but we went through the exhaustive knockout approach where we mutated every open reading frame in the viral genome. And we inactivated all of them and found that it failed to block ISG induction, arguing against viral protein being the inducer of this response. So which led us to think about viral RNA as the trigger of this. And the reason we thought about viral RNA is because there is some complexity to the viral transcriptome that I think most of you appreciate, that HIV transcriptome, which has over 50 different transcripts that have at least been identified, can be broadly classified into 3 different classes. There is the HIV unspliced RNA, which is the 9 kilobase RNA that serves dual roles. It can function as a genomic RNA, or it can function as for translation of the GAG and the GAG-POL polyprotein. There is the incomplete spliced RNA, where only one of the introns is spliced out. And that RNA can serve as for translation of both of the accessory proteins VIF, VPR, and VPU, as well as the envelope glycoprotein. And then there's the completely spliced RNAs, which are very similar to their expression pattern or their splicing pattern to as host RNAs. And they are they're encoding or are translated to TAT, REV, and NEF, right? And so there's also a clear demarcation in the role of REV in the expression of these RNAs. So once these completely spliced RNAs are made, REV is then exported back into the nucleus, which then binds to this RRE sequence in these unspliced or incompletely spliced RNAs and facilitates exports of these RNAs out of the nucleus. So there's exquisite REV dependency for these two classes of transcripts. The reason we were interested in this because this is over 30 plus years of work where there's been clear articulation of how REV functions. REV binds to these sequences, the stem loop sequences in the RRE, and recruits a host factor called CREM1. And this REV CREM1 then is facilitating export of these RRE containing RNAs from the nucleus into the cytoplasm. And once they're in the cytoplasm, they can either be translated or they can be packaged into a new virus particle, right? As opposed to that would be the multiply spliced RNAs where you have the REV independent export, which is facilitated by another nuclear export factor called TAP or NXF1. And those then are responsible for export of the REV independent RRE, multiply spliced RNAs. So if you have reporters that are in place of, say, in this case, NEF, which is a multiply spliced transcript, you can use that as a reporter of multiply spliced gene expression, okay? So that's what I'm gonna use. So this is the data from that initial finding where we had wild type virus. We introduced a mutation in REV called M10, and this is a mutation that was initially described by Mike Malin now 30 plus years ago, which is a transdominant REV mutant that binds to RRE, but fails to recruit CREM1. So the transcripts that have RRE in them get retained in the nucleus, but are not exported out into the cytoplasm. So we are using GAG expression as a readout of that. And so the M10 virus, when you infect macrophages, fails to express GAG in the cytoplasm. You can play some games with this. We introduced another retrovirus constitutive transport element called MPMV-CTE, which can export RNAs in a REV-CREM1 independent manner of unspliced messages. So when you insert those in the sequence, even in the presence of M10 mutation, you can rescue GAG expression in these cells. So that's just a nice control. You can put that CTE element in an antisense manner, and that fails to rescue. You can delete RRE and put CTE back, and you can get the same readout. Okay, so the system works. We can control the types of RNAs that are getting exported out of the nucleus. So we then ask the question, which of these transcripts drives this induction of innate immune activation? So shown here is that same Western blot. And I'm just showing you infection status of these macrophages where we are monitoring establishment of productive infection by GFP reporters. So these are in the multiply spliced site in the NEF open reading frame. So all of these viruses express GFP. But when we look for the CD169 or IP10 induction, the surprise is that only the wild type virus induces it. As soon as we infect with an M10 virus, which is the REV deficient virus, which establishes infection, but fails to induce CD169 or IP10. Okay, so this argued that it's one of those spliced, unspliced transcripts, incompletely or partially spliced transcripts that was driving this. The surprise was that when we rescued that expression using CTE, where infection was rescued, we failed to induce any of these ISGs in these cells. So this suggested to us that there was an additional requirement, which was that you needed REV-CREM1 dependent export of these incompletely or partially spliced transcripts from the nucleus into the cytoplasm to induce these type 1 interferon responses. Okay. Even further, we can create defective proviruses, and I'm just showing you one of these viruses here, but we've created other ones as well, where we've inactivated all open reading frames, just left REV and TAT intact, inserted fluorescent reporters such as tag RFP in the place of GAG, inserted GFP in place of NEF, and we can monitor tag RFP expression as a reporter of unspliced RNA expression in these cells. And we can measure and compare that to a conventional lentivector, where the fluorescent reporter is driven off a intronless RNA using a heterologous promoter, in this case EF1 alpha. So in this case, tag RFP expression in the first case would be Rep-Prem1 dependent. The ZS green from the conventional lentivector would be independent. And the important difference is that tag RFP is from the intron retained RNA export pathway. And we find is that only the lentivector that is expressing the tag RFP from an intron retained RNA is inducing these responses, while the lentivector that expresses spliced RNAs exported in a conventional manner fails to induce these responses. So this, we would argue, is a viral sequence independent induction of type 1 interferon. So RNAs that are exported in a Rep-Prem1 manner, which retain introns, are inducing these responses. Okay, so this was published about 7 years ago, and it was nice to see, but Jeremy Lubin's lab also had very similar findings. We had back-to-back papers, which led to some confidence in this. But for that 7 years since, something happened, so which diverted our attentions. And so we kind of lost focus a little bit and started working on SARS-CoV-2 and other things that happened. And Jeremy's lab as well, we were doing this side by side. And we both were going after all of the known sensors that are present in the cytoplasm. And we went through an iterative knockout, knockdown approaches in primary cells. And I'm going to summarize about 5 years of work in one slide. But basically, this was a way to knock out and knock down almost all of the dead box helicases, as well as other RNA binding proteins which have been implicated in inducing innate immune activation downstream of pathogen viral nucleic acid detection. So this is a summary slide of what was the most important relevant ones. So it's well known in the field, at least, that the cytoplasmic RNAs of pathogen origin can be recognized by the RIG-I-like receptor families, members called RIG-I or MDA5. The RIG-I protein recognizes RNAs which have exposed 5' triphosphates and some double-strandedness, and there are other signatures as well. MDA5 is a little bit more of a mystery, but it's well appreciated that it requires at least 500 base pairs of double-stranded RNA and high complexity. So it has a requirement that is a more rigorous double-stranded RNA than, say, RIG-I. Both of them signal then through an adapter protein called MAPS, which then turns on IRFs and NF-kappa B as well, and turns on type 1 interferon responses. Opposed to the cytoplasmic RNA sensors, there are the endosomal RNA sensors, which are recognizing either single-stranded or double-stranded RNAs, and they require a host factor called UNC93B1 to export that TLR from the ER membrane to the endosomal membrane. And the reason I'm highlighting that here is we take advantage of this. We can knock out UNC93B1, and it mislocalizes all of these TLRs, so the TLR sensing pathway is completely abrogated in an UNC93B1 knockout. So we may knock down, and the other ones that are of interest are PKR or oligoadenylate synthetases, which also can function as double-stranded RNA binding or nucleic acid sensing proteins in the cytoplasm of an infected cell. So shown here is the data from a graduate student in the lab, Sita Ramaswamy, and where she knocked down expression of RIG-I, UNC93B1, or MDA5 in macrophages. Shown here is the RT-qPCR data and the protein where we have knocked down RIG-I, UNC93B1, or MDA5 quite well. And when we establish infection in these cells, we establish similar levels of infection in these macrophages. But when we look for IP10 induction or type 1 interferon expression, only the MDA5 knockout or knockdown is the one that fails to induce type 1 interferon responses downstream of HIV infection in these cells. This is work that was first published by Jeremy Lubin's lab, and then we followed up after that. So again, we were doing stuff together without knowing each other of what we were doing. But it's nice to see that we're able to recapitulate each other's findings. So this is good. So we have identified a sensor. It's MDA5. So what about the rest of the pathway? So this was a lot of work that Sita did, where she's, oh, what is going on here? What did I do? Oh, okay, all right. Do we need that or? Okay. I can keep. It's okay. Don't worry about it. Yeah, don't worry. I don't see anything. Oh, there it is.
Yeah, maybe because you click that. Yes,
That's gone.
perfect.
We don't need translation. All right. So what is the signaling pathway downstream of this sensing? So Sita knocked out individual cofactors here, MABs, TRAF6, IKK beta, TBK1, IKK epsilon, all the IRFs. This was her PhD dissertation. And at the end of it, what she identified was that the pathway required MDA5, MABs, TRAF6, IKK beta, and IRF5, which was also a little bit of a surprise in this because there seemed to be a higher role for IRF5 in macrophages downstream of RNA sensing. I'm just going to show you that IRF data, in that when she was able to knock down expression of each of these individual IRFs, IRF5 seemed to have a more significant role in inducing type 1 interferon responses. Not that IRF3 and 7 were not needed, the surprise was that IRF5 had a predominant role. So this was her finding, and this helped us to identify that there was a unique pathway that was being triggered downstream of HIV RNA expression in macrophages. But this also opened up a lot of questions, and those questions are still under consideration. And one of them, and this is findings from lots of investigators in the field. This is from Sun Her's lab at Harvard Medical School, where they clearly identified a requirement for MDA5 as a binder that binds to a double-stranded RNA fragment of a certain size, which then recruits additional MDA5 monomers to form this fibril-like structure. Okay, so that's the model where the monomer initially binds to a double-stranded RNA molecule, approximately 500 base pairs in length, which then recruits additional monomers to that fibril, creating this large RNA-like structure where there's both MDA5 inter-MDA5 interactions or intra interactions, which are then stabilized by TRIM65 mediated ubiquitination. The challenge is that it's what has been thought is this 500 base pair to 1,000 nucleotides requirement for double-stranded RNA for these MDA5 recognition machinery, which, as far as we know, do not exist in the HIV-1 genome. There is no 500 base pair to 1,000 nucleotide double-stranded RNAs. We looked. So this is work by Salil Jalloh, a postdoc in the lab, who was then looking for these double-stranded RNA epitopes. And we used an antibody called J2, which some of you might have used as well. And we had to look hard, and we find some double-stranded RNA positivity in the cytoplasm of an infected macrophage. It's not much, but it's there. It's not seen in an M10 virus infection, and it's obviously not seen when you block infection with an RT inhibitor. But J2 specificity is not just 500 base pairs to 1,000 nucleotides. The epitope actually has been mapped, and it can recognize as small of a region as a 15 base pair double-stranded RNA. So it's not really clear if these positivity that we're seeing is truly large double-stranded RNAs, or if they are actually viral of origin, because we were not able to clearly identify if this positivity correlates with viral RNAs in the cytoplasm. So the challenge here is that the HIV genome architecture, and this is shape data that was published by Kevin Weeks over 15 years old now, which have argued for different stem loop enrichment at the 5' UTR, the GAC-POL frameshift region, the RRE, the 3' UTRs. There are these regions in the genome which have been thought to have higher secondary structure than other regions of the genome. And some of these structures actually have been identified, have been solved using NMR. And this is work done by Mike Summers and Ella Stelznitsky. which have focused especially on the 5' UTR, and have shown this exquisite structure to this 5' UTR. Shown here is actually the genomic RNA, which is double-stranded RNA, which has this open dimerization sequence or the kissing loop. And shown here is the two RNAs that are in close proximity to each other. So we were intrigued by these structures, and we thought, are there some of these RNA structures that are contributing to this MDA5 recognition? The other questions we had was whether intron retention was a requirement for this, and whether there's density or dimerization or intracellular localization of these RNAs were all critical aspects of MDA5 requirement. And there could be others as well. So we went about this by looking at those unspliced RNAs, and especially on the 5' UTRs. The reason we were interested in is that there's some recent literature that has argued that there are two functions which these RNAs are responsible for, and there are two different forms of RNA that perform these functions. So as I mentioned to you at the start of this talk, unspliced RNAs are required both for translation of GAG and GAG-POL, but they're also packaged as a genome, right? So how are these two opposing RNA functions controlled? So it turns out viral transcription is not initiated at a single site. There's some stochasticity, and that there are two different guanosine residues that are where the RNA pol 2 can start and form two different RNAs. So there's a stretch of 3 guanosines at the start of the R sequence, and the 5' proximal guanosine, if the RNA pol 2 starts from there, makes an RNA called Cap3G RNA. If the RNA pol 2 starts transcription from the distal guanosine, that one makes the Cap1G RNA. And the difference between those two RNAs and across the entire 9 kilobase genome is the 2 nucleotides at the 5' end, and that's it. Okay, so and this transcription start choice seems to define the structure and function of these unspliced transcripts. And this is work from Wei-Su Hu, Alice Telzhnitsky, Mike Summers, Ben Berkut, lots of investigators in the field who have done seminal work in this space. So to summarize that work, Cap1G preferentially serves at the viral genome because the structure that it forms, it has this coaxial stacking of the TAR region and the poly A, which exposes the dimerization sequence. And that allows it to interact with the second RNA, the second genomic RNA. So that is a requirement for the viral packaging. The consequence of that is that the 5' cap structure, all of these, by the way, all these RNAs are capped, that 5' cap structure gets sequestered in this form of viral genomic RNA. So this RNA is less likely to be translated, but it doesn't mean that it's not translated. It's just less likely. As opposed to that is the Cap3G RNA, which, because of the extra guanosines, follows a completely different structure, where now the poly A sequence and the TAR don't coaxially stacked, but the consequence of which is the dis or the kissing loop sequence gets sequestered. So it cannot bind to the second dis sequence in the other RNA. The other consequence of that is that the cap on this form of RNA is exposed perennial all the time. So this means that this RNA is predicted to be the more translationally competent than this form of RNA. So this RNA is not utilized for packaging, but is used for translation. Just remember, this is not a black and white. This is a probability event because all of these RNAs have an ensemble structures. They keep flipping and flopping between structures. So this is the model that Cap1G can dimerize and get packaged, while Cap3G gets translated. So Ivy Hughes, a graduate student in the lab, asked the question whether these two forms of RNAs that are expressed in the cytoplasm contribute to differential MDA5 activation. We took advantage of some of the mutants that Alice Telzhnitsky's lab has made, where they're able to not only to generate a mutant where they inserted 2 nucleotides in the U3 sequence that forces RNA pol 2 to only transcribe from the distal guanosine. So in this case, this virus only makes Cap1G, or another virus where they inserted 2 nucleotides, which makes the virus only initiate transcription at the 5' guanosine. So you only have 1G only or 3G only viruses. So shown here is the macrophages infected with those viruses. You have the wild type virus, which makes both. 1G, which makes only 1G, Cap3G, which makes only 3G. They're all competent for single round infection. It's not the spreading infections which are the problem, but we don't need to worry about spreading infections in this case. So when we look at induction of interferon responses, the surprise was that only Cap1G induced interferon responses. Cap3G, which, as I will stress again, only differs by 2 nucleotides at the 5' end of the RNA and nothing else, and no differences in the open reading frames, no differences anywhere else, fails to induce those type of interferon responses. Is it MDA5? So we knocked down MDA5, RIG-I, or MAVs in macrophages and did the same experiment where we infected with these viruses. And we measured, again, the ability to induce type 1 interferons and find that knockout or knockdown of MDA5 or MAVs, but not RIG-I, knocks down type 1 interferon responses. So Cap1G expression is being sensed by MDA5 and MAVs in these cells. Are they directly interacting with them? We overexpressed an epitope tagged version of MDA5 and did immunoprecipitations of the different forms of these RNAs in the cell, and found that only the unspliced form of RNAs, either the wild type or the 1G, can be immunoprecipitated by MDA5 flag tag version, but not the 3G RNA or infected cells in the presence of RT inhibitors. So RNAs that are being present in an infected cell cytoplasm can be immunoprecipitated by MDA5 of the 1G variety. And the controls are the multiply spliced RNA does not get immunoprecipitated, or a control RNA, in this case, actin B. So again, we are convincing ourselves that indeed this is MDA5 dependent and that it's recognizing Cap1G RNA. So what we have now is that we have 2 forms of viral unspliced RNAs, one which is immunogenic, which we think seems to be necessary for packaging and assembly, and one which is responsible for translation, which is immunosilent, even though their RNAs are not that different. And we think it's primarily, so there's 2 requirements at this point that were left. One was cap sequestration or the dimer competency, as I measured these 2 RNAs dimerized before they're assembled. So we took advantage of additional mutations that Alice Telznitzky and Mike Summers had recently defined. They were able to induce our mutations within the polyase sequence, which stabilized that stem loop structure, so that now it coaxially stacks with the TAR sequence. So this is a 3G with the mutations in the polyase sequence. And now the structure looks somewhat similar to the Cap1G RNA, with the exception that the cap on this RNA still remains exposed. So the difference between this mutant Cap3G that I'm calling and Cap1G is that the structures look identical except for the cap accessibility, where in the 3G mutant, the cap still remains accessible, while in the Cap1G, the cap is buried. All right. So we asked the same question again, whether they induce interferon responses. And the surprise was that the structure that looks very similar to 1G again fails to induce type 1 responses. This would argue that the dimer competence form is not the driver, but rather the cap that is exposed, or whether cap is sequestered, is what is triggering these responses in an MDA5 dependent manner. All right. And finally, is this still RevCram1 dependent? Because I started this discussion by arguing that there was a unique nuclear export pathway that was needed for this. So we made the same mutants that I described to you before, the M10 construct, or a 4X CTE, which was inserted in place of POL to rescue M10 export. And we find that, again, when we look at these questions, ask interferon responsiveness, only the 1G wild type, but not the M10 or the 4X CTE rescued form of MG fails to induce these responses. So at this point, what we have convinced ourselves is that MDA5 engages this Cap1G RNA, which leads to MAV's activation and type 1 interferon responses. We think 5' cap sequestration is needed for this, and that nuclear export pathways, and specifically the REV-CREM1 mediated export, is critical for this. And one of the intriguing findings from this, and which we are following up and quite intrigued by, is this transcription start site heterogeneity, which has been described for all HIV-1 isolates, as well as some SIV-CPZs as well. And we think maybe this is a virus-encoded mechanisms to modulate those innate immune responses. So the right combination of 1G, 3G RNA expression is needed to control or prevent over-exuberant type 1 interferon responses in infected cells. And that if you only have one form of the RNA, you have problems. But that's still work in progress. The other part of this is that, as I mentioned, we also see age-associated intrinsic differences in these RNA expression and differences in expression of various adapter proteins that are required for this induction. I didn't show you that data, but we find that IR5 is specifically upregulated in aged macrophages. These are people who are 50 and above. Sorry, I'm calling 50 plus as aged, but and for 35 and less is younger. And we find that those who are 50 and above, their macrophages, their monocytes constitutively overexpress IR5. And we find that RNA or HIV infection of those aged macrophages leads to a higher cytokine output per cell on a per infected cell basis. So we think there is an aging comorbidity argument that can be made in this RNA sensing argument. And what we are now specifically interested in is the question of whether this latent reservoir that remains transcriptionally competent can contribute to this chronic inflammation because of this pathway that is present in these latently infected cells. Okay, I'm gonna switch gear and finish up in the last 10 minutes with the description of the last step in the virus life cycle, assembly, and another innate immune sensor that we've recently made some descriptions of, and that is what detects the viral protease activity. So for this, I'm going to describe another sensor, and in this case, I'm going to describe to you the inflammasomes. So inflammasomes are just like PRRs, like other TLRs or RLRs. They can recognize PAMs in the cytoplasm of an infected cell, but what's different about them is also they can also detect pathogen-encoded activities. So this is a new area of innate immune research, which is focused on pathogen-encoded activities as what are driving these sensing mechanisms. Effector-triggered immunity is the name for it. And what happens is that the consequence of that is that these inflammasome components, for example, these monomers, upon detection of PAMs or activities, come together and form these large oligomeric structures in the cytoplasm, and that activate this inflammatory protease called caspase-1, which then cleaves its targets like IL-1 beta and gasdermin D, which lead to inflammatory cytokine secretion, as well as pore formation in infected cells that results in lysis and biopyroptotic mechanisms. Okay, so this is a very effective way of clearing infected microbe-infected cells. What we got in, the reason why we got into this question was that there has been some really seminal findings in the literature that has argued that there is this arms race between viral proteases and a specific inflammasome called CARD8. Turns out CARD8 inflammasome detects viral protease activity from HIV-1, SARS-CoV-2, picornaviruses, and they do so because they have this very intrinsically disordered N-terminus loop, which has sequences which mimic viral protease cleavage sites. So detection of viral protease results in cleavage of this disordered loop, which frees up the C-terminal domain of CARD8 so that it can oligomerize, form a large oligomeric complex, and initiate pyroptosis in the cell. Right? So this CARD8 is rapidly evolving to sense a diversity of viral proteases. And as you can imagine, viruses evolve in response and are much better at it. But we were intrigued by identifying what methods HIV has taken on to conceal its proteolytic activity from CARD8. Okay. So this work got started about 4 years ago, and this was a paper by Liang Shan's at WashU, whose lab identified that CARD8 detects HIV-1 protease activity leading to pyroptosis. And they did by using a trick. They take infected cells that were already infected, and then add in non-nucleoside reverse transcriptase inhibitors, NNRTIs. So addition of NNRTIs in infected cells causes GAG and GAG-Pol dimerization. So you require protease activity. In order for protease to get activated, it has to dimerize. So adding NNRTIs to the cytoplasm of an infected cell enforces GAG-Pol dimerization, which reveals protease activity, which then is detected by card8, and the cell dies. Okay, this is a trick. So normally it does not happen. But then last year, again, work from Liangshan, and now also Patrick Mitchell and Michael Emmerman show that incoming virus particles, that is, especially under conditions of high MOIs or cell-to-cell transmission, where you're delivering large amounts of viral capsids into the cytoplasm, the associated protease that's present in the incoming virion is able to activate card8. The argument here was that this was a mechanism that was proposed that might contribute to the bystander cell cell death that has been observed reproducibly in pathogenic lentivirus infections. That's a model that's still being investigated. But what we were thinking was that there's these kinds of findings then would argue that during virus assembly, before virus has completed its life cycle, there must be methods or innovations that the virus has used to conceal PR from being detected by card8. If it doesn't, if there is premature protease activation, that would result in cell death and a loss of producer cells, which would impact viral fitness. So this is work again by Ivy Hughes, who's got 2 different projects in the lab, and this was recently published. So I don't need to go through this assembly process, but all I'm going to tell you is that there's recruitment of GAG and GAG-Pol monomers to these membrane microdomains at the plasma membrane, where they find their dimeric RNA and where they assemble. And the whole goal here is that there's only 5% of all GAG is GAG-Pol. So there's very little GAG-Pol monomers in an infected cell cytoplasm, which remain far from each other. They are prevented from being dimerized. And this only happens right at the time when the virus particle is either cleaved so that it by the escort complex, so it buds out, or after budding. And that still remains a bone of contention in the field. It's not clear when protease is being activated, maybe right at the type of budding or after budding. But regardless, it's concealed all the way through all the steps of the virus assembly process. So the other key point is that proximity is what drives the dimerization. And this is still unclear as to what about the membrane assembly site and what are the requirements of the environment, but that forces the GAG-Pol to come together so that protease can get active and mature the virus particle, and that still remains unclear. But we were interested in seeing whether indeed different GAG microdomains are controlling this card8 or controlling PR activity, and in essence, preventing card8 activation. So we made gross deletion mutants to get at this question, and I'm going to show you data from 2 of them, Matrix and P6. So the model system that we use for this study is that we use single cycle viruses that infect either THP1 macrophages or primary CD4 positive T cells. In macrophages, we may look for IL-1 beta secretion as the measure of a pyroptotic event or inflammasome activation. In the case of activated T cells, we do these infections in the presence or absence of viral protease, and we measure the survival of the cells as a measure of card8 activation. Okay, so in this case, presence of a protease inhibitor should prevent cell death, and we combine that and report that over the death that is reported in the absence of protease. Okay, so the reason we have to do this in CD4s is that they don't make IL-1 beta, simplest time. So the mutations that we did were matrix deletions, which was a gross deletion, all the way from amino acid 6 to 125, that just retains the myrcillation signal so that membrane targeting is still retained. We deleted the zinc finger of the nucleocapsid, or we just mutated the mutated the PTAP sequence that is required for recruitment of the escort complexes to facilitate release from facilitate budding from the infected cell. So shown here is the data. Again, lots of repeats done many times. So focus your attention on IL-1 beta first. So here's uninfected, wild-type infected, where you see some background IL-1 beta secretion. You delete matrix, and you see a robust induction of IL-1 beta in this case. You remove protease from that delta matrix, and that induction of IL-1 beta is lost. You prevent infection with efavirenz pre-treatment before infection is established and IL-1 beta secretion is lost. You prevent membrane association of that matrix-deleted mutant, and it's substantially attenuated in IL-1 beta secretion. You introduce capsid mutations that prevent oligomerization of the GAG that is recruited to the membrane site. And that, again, loses the ability to induce IL-1 beta secretion. You delete NC, and that has no impact on this. You just mutate the PTAP domains, and now you again see robust IL-1 beta secretion, which is prevented by addition of a protease inhibitor. So lots of data. Just to summarize here, matrix, when you delete it, which was a big surprise, activates IL-1 beta secretion, suggesting that PR activity is being revealed when matrix function is being tampered with. And you can prevent that by preventing oligomerization by capsid mutations or by preventing plasma membrane association with that. So a unique spatial localization of GAG and enforced GAG polymerization by matrix mutations induced CARD8 activation. P6, on the other hand, is a different argument, we think, and I'll show you the data and I'll explain what that is in a minute. So we think this is CARD8, and we know it is CARD8 because we can knock out or knock down CARD8. This is knocked down in THP-PMA macrophages. We did the knockouts in primary CD4 positive T cells using CRISPR, the same phenotype. You knock out CARD8, the delta matrix and the delta P6 fail to induce any IL-1 beta. So this is CARD8 dependent and PR dependent. We see the same thing in T cells. This is wild type primary CD4 positive T cells infected with wild type virus. And I'm showing you the cell killing assay here. And there isn't much. Obviously, cells don't die because of pyroptosis when everything is functioning normally. You add an NRTI and you force GAG polymerizations and the cells start dying. You infect with a matrix deleted virus, the same one as before, the cells start dying. You disable PR activity in the context of matrix, and there is no more cell death. You infect with the P6 deficient capsid and then a GAG, and you again see cell death. So same phenotype as THPs, macrophages as this. And what we think that's happening is that when there is matrix deletion or when there is PTAP attenuation, we find that the virus particles that assemble lose some of their enzymatic activities from the virus particles. So in this case, either RT is what we are using because the antibodies are good. The amount of virus that gets pulled out of that gets released from the cells has either no enzymatic activity in the case of delta PTAP. So that means that the PR or the RT is getting diffused out from the assembly site back into the cytoplasm to trigger CARD8 activation. Or in the case of matrix, there seems to be premature GAG-POL loss. So it's somehow dimerizing prior to assembly getting completed. So this is a model which still requires quite a bit of studies, but we think that there is premature either protease activation in the context of delta matrix or protease release from the assembly site that causes CARD8 activation in the case of delta PTAP. Okay. The reason we got super excited about this is because there has been an evolutionary argument for control over CARD8 activation. So the chimp viruses, the ancestors of HIV-1, which infect chimps, do not recognize the chimp version of CARD8. So the CARD8 that's in the chimps is unable to get activated upon SIV-CPZ PR or HIV-1 PR expression. But the human CARD8, so when the ancestral virus jumped into the human species, it encountered a CARD8, which has the capability of getting activated by chimp viruses or HIV-1 PR. This is again work from Michael Emmerman and Patrick Mitchell. who showed this interesting phenotype. And there's actually a single amino acid difference in the cardate recognition site for the PR cleavage site in chimp versus human. So when HIV-1 jumped into humans, it now had to deal with a novel immune pressure. So we argue that the matrix has evolved in the course of HIV-1 infections to modulate PR activity and cardate sensing. And the reason we think this is the case is that there is this unexplained finding in the literature for now over 20 plus years, which has shown that there is a unique amino acid that is found at position 30 in matrix, and it's universally methionine and leucine in all extant SIV-CpZ isolates. And it's universally arginine or lysine, a basic residue in all HIV-1 subtype B isolates. This is extremely well conserved. This is work from Beatrice Hahn, Paul Sharp. We argue that the SIV-CpZ, because it has a residue at a basic methionine leucine at this hydrophobic residue, could care less because cardate protease recognition it didn't care about it. But that as soon as it jumped into the human species where cardate became an issue, it had to mutate to an arginine or a lysine, which would have allowed it to evade cardate in the human population. This is again a model. So I want to prove this. So the way we proved it is that, we partially proved it, is we made replication competent or single cycle viruses where we substituted just the chimp matrix sequence into the HIV-1 matrix, or we just mutated that single residue from lysine back into methionine. And we did the same cytotoxicity assays that I just described to you. So the killing assay with the chimp chimera is greater than the wild type HIV, or the single point mutation is by itself sufficient to increase cytotoxicity in primary activated CD4 positive T cells. The effect sizes, as you can see, are much lower than what I showed you for the gross deletions. This is much more subtler. And that has also been observed even with these different mutations that have been described, where there are small increases in replication fitness for this matrix mutant that expresses lysine instead of methionine. So we think this is the reason why there is an evolution to this sequence signature in matrix to evade the cardate detection. How, why, and how does it evade that process is still something that we are actively investigating. But we think that HIV matrix, which was unappreciated until now, that it controls protease activity, and it does so by controlling where the virus assembles and controlling how GAG-Pol dimerizes at this assembly site to prevent premature PR activation. And that this near universal adaptation that existed has to have happened to help evade the immune pressure that cardate enforces. Right. So I'm going to end by actually thanking the people who did the work. I highlighted Ivy Hughes, who has led 2 of these stories that I've described. She's a student who's going to be defending her dissertation in the next month. Sita Ramaswamy, a previous PhD student, identified MDA5. Salu Jalot has been involved in a project that I did not describe to you on another inflammasome called NLRP1. This is a collaboration, long-standing collaboration with Hisashi Akiyama at BU. We work closely with Andy Henderson and Manish Sagar on the clinical cohorts. Alex Telznitsky at University of Michigan on the 1G3G RNA story. Worked with Zandria Ambrose on nanoparticle stuff, which I didn't get a chance to talk to you about, as well as Bjorn Reinhardt, and Christine Cheng and Sanjay Tyagi on the iPSC microglia systems, and funded by all of these sources, especially NIH, who have funded a lot of this work. Thank you.
So questions?
Thank you. So very interesting. So I was wondering, do you think the mutations in matrix can alter the dynamics of the GAG on the immature particle or on the budding so that they will interfere with the GAG pole finding each other to make the dimer, or is that
Yeah, that's the model we have. We haven't proven that, but the thought is that the matrix mutations, especially these basic amino acids, which are known to interact with negatively charged lipids like PIP2s. This has been a finding that Eric Reed and others have made in the lab, in the field, governs the assembly site choice. So when you make changes to those basic residues, you are subtly altering where the virus particle assembles. Because of the change in membrane microdomain, we think that that changes the GAG-Paul dimerization kinetics or probability, which triggers the PR activation. But that's a model. We don't have.
But do you think it depends on the site where it buds, or is just wherever it would bud, you will have been altering the dynamics?
The speed is also a possibility. So those are both possible. Not only is the site presumably altered, but that the speed at which the assembly progresses as well is also ordered. So both could be at play, and we leave it at that, saying it's a spatial temporal control, because we don't have a definitive answer that proves one way or the other at this point. But that's something that we're doing right now.
But yeah, thank you. That was very interesting. I have a few questions. So what happens if you only overexpress the protease? Do you trigger IL-1 beta?
Cells die.
The cells die. And with the CAP3G versus the CAP1G, I found this very interesting, but it seems that the CAP3G is impaired in its replication or in its fitness. Do you think that might be sufficient to explain some of the observations that you made? Because, well, there is a small decrease in infection levels even.
You picked, that's great. So CAP3G actually fails or is quite attenuated in its ability to package RNA. So in a spreading infection, it has a very poor fitness. So single round infections, you can make HEC293 transfected viruses, which is what we do, and you can dump in enough MOI to overcome that initial inhibition, so you get single round infection established. In the next round, the RNA is not capable of getting packaged, so you have severe deficiencies in spread. So that's what's happening. But in that cell, the RNAs are still getting expressed. It's just not getting packaged. So CAP1G, on the other hand, has no such problems. It's getting packaged. It's getting viral proteins are getting translated, but there's clearly pressure to maintain both transcriptional SART sites in an infected cell, because we think that CAP1G is just over exuberant in its responses. So that's probably why we think there's CAP3G requirement. But again, still dependent.
And I have a last question regarding the first part. I found, well, your findings are very interesting regarding MDA5, but there are other sensors like SIGA, Sting, or even TLR7, 9, which are known to sense HIV in macrophages. So what are the specificities in your system in which you only see MDA5 finally?
Yeah, that's great. Thank you. Thanks for reminding me of that. And I forgot to mention that. So in regards to TLRs, I think those were only seen when there were infections done in a cell-to-cell infection manner. That was one thing, and I think it was restricted to PDCs most often than not. And I don't know if that ever was seen in the context of a macrophage or a CD4 positive T cell. So that I think that might be a cell type specific requirement for TLR 7/8. In context of CGAS Sting, I think that I think this remains controversial, but I think most of the CGAS Sting requirements are sensing are only seen when there's defective virus particles that are present. So in the sense that there's capsid that's compromised, or there's ways to inactivate or mutate the capsid using small molecules as well too, which either promote accelerated capsid disassembly upon infection that reveals the nucleic acids or reveals the RT intermediates. In the context of productive infection with an infectious virus that has an intact capsid that uncoats optimally in the nucleus, I don't know whether there is any CGAS detection of that RT intermediate. that's our interpretation. What I showed you today was all about late. I didn't show you any day 1 or day 2 ISGs. We looked. We don't see that much on day 1 and day 2. So that's where we are. Day 3 and beyond.
Yeah. If I can add something, yeah, we are completely agree with you because we tested gas and we could not get any activation except if we disassemble the HIV amylose in the nucleus. In that case, we have the exposure of the double-stranded DNA. Otherwise, yeah, there is not.
Hi, thank you very much. Maybe I missed the point, but for me, it's not clear how the different 3G versus 1D cap RNA are delivered. I mean, you see, because it's a random process or it's an active process that deliver just the 1G at the assembly site. And this goes with the following question, that is, this means that you need the interferon response is induced at the assembly site, because there is the one that induce the interferon response. So do you see colocalization or, I don't know, some MLO in which the immune response are induced?
Yeah, that's a great question. Okay, so the way we did the experiment is that Alice made these mutations in the LTR, which allowed us to only express RNA that has a Cap1G structure. Okay, and the way that was done was that there were 2 nucleotides that were either deleted or 2 nucleotides that were inserted in the U3 sequence. So that allowed for those 2 different forms of RNAs to be made. Okay. And then these RNAs were then transfected into, these proviruses, plasmids were transfected into 293s. You collect viruses, so they only make one form of the RNA and infect macrophages with that. So then we control which RNA is getting expressed in the infected cell. So that was our delivery method. So none of these were directly transfecting RNA into the cell, but these were all dependent on infection establishment, de novo viral transcription of that RNA, which then triggers this response. In regards to the second question, yeah, so is this an assembly site question, assembly site sensing again? So as you mentioned, the Cap1G is the one that dimerizes, and that's the one that gets picked up by the viral particle as it assembles at the plasma membrane. So it's very likely that you might have a sensor that's sitting there to detect that versus the Cap3G, which never makes it there, and it's only already getting translated. That's an open question. I don't know the answer to that.
I don't understand why the 3G never get there.
Oh, so the dimerization sequence in the Cap3G is not exposed, so it doesn't.
Yes, but
Oh yeah. Oh, I see. So all of these RNAs, you bring up a great point. All these RNAs go everywhere. It's just that whether they are retained at the membrane assembly site or not is the question.
Yeah.
So they constantly circulate. So I think there's an accumulation of the dimerized form of the RNA because of GAG nucleation on that RNA at the plasma membrane assembly site.
Okay.
So that causes it to retain there versus the 3G, which constantly is moving and it never gets dimerized, so it never gets trapped by GAG, presumably.
Yeah, I have a question about that. You show nice imaging about the antibody that recognized double-stranded RNA, but not with the M10 virus. Did you try to do a co-EF with the MDA5 antibody to see if
Yeah,
localize?
that's a difficult experiment. MDA5 antibodies are not very good. I was talking to Anastasia about that, but it's a very challenging experiment. We've tried; it's not working very well.
And by MDA5 GFP, for example,
so overexpression means then that MDA5 goes everywhere.
Yes,
so the problem then becomes is that you're hard to press to tell what is real and what's not real. We would like to do this in a considerably expression.
I have to thank you. It was really nice. I have 2 questions. So one is a little bit like Olivier about all the sensors in macrophages, and well, I was a little bit naive because I thought that there was nearly no sensing in macrophages, and it's not what you're showing. But I knew about SIGAD and everything. I thought you couldn't do innate immune activation in macrophages. So then I come up with this question: what about CD4 primary T cells? What do you think? Could we trigger some kind of sensing if we look differently in these cells?
Yes, that is correct. So I think we are struggling to identify if there is a sensing event in T cells because we don't know what to look for. Right. So the reason we all do stuff in macrophages is that there are ways to detect that response, like type I interferon, like IP10, like a cytokine, or something else which is clearly visible and it has a very high signal to noise ratio. In primary T cells, we don't have good markers to identify, or we have not identified good markers which tell us whether there is a sensing event happening or not. Jeremy has argued that you can use ISG15 or CD86 as, sorry, not CD86, ISG15, and they do intracytoplasmic ISG15 expression, and they see a response in CD4 T cells to the same RNA. So he's published that. But yeah, it's a challenge. But I think the sensing event remains the same. This is not a macrophage specific phenotype. This is my bias, by the way.
And from your results, well, as you said, MD5 is supposed to sense double-stranded RNA. So here it's a secondary structure of the HIV RNA. So, but can you deduce that MD5 is sensitive to something more than double-stranded RNA? Is there something more you could say about the mechanism of the sensing?
Yeah, no. So that's a big one, right? Because the structures that I'm showing you will probably exist on eukaryotic RNAs as well. I mean, these are not big stem loops that I'm sure there are some eukaryotic RNAs which also have these stem loop-like structures. So there has to be mechanisms that are in play that prevent MDA5 from getting activated by host RNAs. Otherwise, it would be autoimmune disease. It would be just a walking ball of cytokines. So that's not possible. So there has to be a host proteomic requirement, which is different. There has to be a localization that has to be required. There's probably a threshold requirement, not probably, I know there is a threshold requirement that requires a certain amount of RNA that's present in a unique environment that triggers that response. In the absence of those, we think not. I think that a reason why 3G doesn't do it is because there's a translational argument. So maybe if it's shunted to the translational compartment or getting translated, that RNA is no longer available for this sensing pathway.
Oh, last one, very, very short. Have you tried to look at auxiliary proteins, whether you have sensing or not?
Ordinary protein?
Viral accessory proteins. If you infect
accessory proteins.
Accessory proteins. Yeah. Sorry.
No. Yeah, we knocked all of them. No difference. Nothing, as far as we can tell. I think there is a paper, I think from Frank Kirchhoff's group, that has argued that it's NEF. So, and there's somebody here from Frank's lab who can talk more about that. But they've argued that NEF controls that activation.
Hello. You mentioned that for sensing, you needed to go through that CREM1 dependent pathway. So can you speculate what is special about this pathway in terms of localization, cofactors?
Yeah. So I don't have an answer for you, Lisa, but what we are doing right now is a proteomic screen. We are IPing the proteome associated with the REV-CREM1 exported RNA versus a CTE exported RNAs to basically get at the question that we're trying to answer, to see if there's any differences. And if there are differences in that proteome, maybe they'll tell us that is a cofactor that is needed for this MDA5, but we haven't done in process.
Okay, just a comment. Thank you very much, because it was very nice to see the story, like we showed for the early infection, how it's going with the reguide sensing on the endogen sensing, and now you show this nice story with MDA5. Actually, it comes to your comment on the translation with the 1G and 3G. I was wondering, have you tried to measure the efficiency of translation whether it helps, if you compare 1G, 3G, because ribosome is really melting, and that can explain everything.
Yeah, that's what our hypothesis is. So we did do that. So the 3G RNA is much higher translational potential. So it makes much more GAG-POL than the 1G. So we think that's what's happening, is that the translational apparatus is inhibiting or preventing that RNA from
melting, because you don't have any more.
Right.
But I was wondering, there is no ARS or something like that, IRS structure, because it looks like IRS, but I didn't follow the.
There's no iris.
No, it's like cap dependent classical, because I remember hearing there was something like IRS like mechanism for the HIV.
There is a GAG. There is an iris in the GAG sequence. I think not everybody buys that argument, but I think if you look at it hard enough, there will be some. Sure.
Okay, thank you very much.
Hi, nice talk. So following the translation issue, are you sure, or Alice showed, that the transcription of these two variants is similar?
Yeah. Oh, that's what the reviewers asked. That's what we're doing right now.
Yeah.
The RNA abundance is different because, as I mentioned, because of the translation, some of the RNA gets turned over. So they want us to do basically a temporal analysis over long periods of time and make sure that we have similar RNA levels. As far as in a single time point, there is about a 4-fold difference between 1G and 3G. So
that can explain the
differences. So
also the structures, it seems like in one of them, the TAR is really not accessible to recruit the transcription machinery. I mean, at least
that's unlikely to be the case, because when you measure GAG expression in the cell, it's robust, if not greater, for the 3G versus 1G.
Right, but downstream genes?
Downstream?
Like, below, like NF or...
Oh, no, but this is protein, right? So this is the unspliced RNA that it has to make the entire...
No, I'm talking on a level of transcription.
Oh, level of transcription. Well, I think we are using protein as a pseudo measure of equivalent transcription. So maybe that's not fair. Maybe we should do RNA. But if we think there's equivalent or greater amounts of protein, to me, that would argue that...
Yeah, you're right. But
that's what we're using as a measure. But fair. I mean, you have to do the RNA too.
Another question.
Because we have time, I'm very interested because you commented that you need a threshold to induce this sensing. And actually, I was wondering, but I didn't know I could ask, why it takes so long, because 3 days is quite long. So my question is, the threshold is determined by the total amount of RNAs or only the 1G?
Yeah, I think it's...
And because the 1G should, so it should be I don't know how it works.
No, that's a great question. First, I'll answer about the 3 days. These are macrophages, so a lot of them, I think this is also work from Francesca's lab, as you require 24 to 48 hours for reverse transcription to be completed and integration to occur, and that there is enough RNA. Yes, that's why it takes 3 days, nothing more than that. But the amount is not only 1G, but you're right, that newly transcribed RNA needs to be built up in the cytoplasm in order to be there for sensing to occur. So I think that's the threshold I was getting at. But you're right, so there is a balance. Some of that RNA will escape, is getting packaged into a new virus particle, and it buds out of the cell. So there is a balance there, and I don't have a good answer for...
Something that goes faster and faster during viral replication.
I don't know.
It goes better and better.
Yeah, I don't know if there are any data out there that has looked at the temporal differences in viral transcription at later time points. I just don't know if that data exists. We should do that, but yeah, maybe.
And I have maybe the last question about IRF5 that you mentioned that is increasing, the expression is increasing with the age. So because there is more senescence in in people affected with HIV, Do you know if there are some studies measuring the expression of IRF5 from people having the same age, infected or not with HIV?
Right. Okay. No, so it's the same whether they're plus or minus HIV. It's only age alone makes a difference. So
So
for
there is not an increase of senescence in macrophages from these people that can explain. So
no, it's more. I think it's more to do with the epigenetic control of expression of some of these IRFs or other things. I think there is associated chromatin access and control of host transcription is leakier as you get older. So that leads to a higher responses from some of these pathogens. I think that's all it is. Turns out IRF5 is one of the factors in this pathway that's upregulated. There are other pathways that have also been postulated to be altered with age as well too. So it's not pathogen induced. It's just the luck of the draw. As you get older, you are much more susceptible to a lot of things.
okay.
You get old.
Okay, I think that we can conclude here.
Okay.
Thanks a lot for the great talk.